Somewhere on the Great Plains, around the year 60 AD, a Lakota healer pressed a strip of bark against an infected wound. The wound closed, the fever broke, and the patient survived. In 1962, the United States government decided that bark had no place in medicine. With the stroke of a pen, centuries of Lakota healing knowledge became illegal. But in 2019, a team of researchers published findings that stopped the scientific community cold. The bark worked, and it had always worked. The people who knew that had been silenced for 60 years. To understand what was lost, you have to understand what existed. The Lakota Sioux inhabited one of the harshest environments on the North American continent, where winters dropped to 40 below zero. Summer droughts cracked the earth, and open grasslands stretched hundreds of miles in every direction with nothing to break the wind. In this landscape, one plant appeared everywhere, a tree the Lakota called Kampa and biologists classify as Prunus virginiana, the chokecherry. Lakota healers, known as Pejuta Wicasa, stripped the inner bark, dried it, and brewed it into a tea for infected wounds, respiratory illness, and fever. They also applied it directly as a poultice to open wounds. This was not guesswork. Over 2,000 years, the Lakota developed a precise pharmacopoeia, an actual system of plant-based medicine tested across generations of patients, refined through observation, and passed down through oral tradition. Ethnobotanist Melvin Gilmore documented Lakota plant medicine in 1919 for the University of Nebraska. His record showed that the Lakota used over 200 distinct plants for medicinal purposes, including chokecherry bark specifically for infection and wound healing. The compound responsible for much of this effect is prunusine, a cyanogenic glycoside found in concentrated amounts in the inner bark. When processed correctly through drying and brewing, prunicin breaks down into compounds with documented antimicrobial and anti-inflammatory properties. The Lakota didn't know the word prunusine, and they didn't need to. 2,000 years of observation had already told them everything they needed to know about what this plant could do. But knowing something and being allowed to act on it, those are two different things entirely. When European settlers arrived on the Great Plains, they brought their own ideas about medicine, and most of those ideas were wrong. In the 1800s, mainstream Western medicine relied on bloodletting, mercury, arsenic, and purging. The germ theory of disease was not widely accepted until the 1880s, and antibiotics wouldn't exist for another 60 years after that. Meanwhile, the US government was running a systematic campaign to eliminate indigenous culture. Native American boarding schools, established by the Indian Civilization Act of 1819 and expanded dramatically after 1870, separated children from their communities and their traditions. Healers could no longer pass their knowledge to the next generation in the traditional way. Children were punished for speaking their languages, practicing their traditions, or acknowledging that plants like Kampa had any value at all. Some knowledge survived in the records of outside observers, including missionaries, anthropologists, and soldiers. But their documentation was fragmentary, filtered through a cultural lens that treated indigenous medicine as primitive superstition rather than clinical knowledge. By the early 1900s, the chokecherry bark compounds began appearing in commercial patent medicines, sold in bottles and marketed to white Americans as folk cures. This was not a celebration of indigenous knowledge. It was extraction without credit. The Lakota received nothing, and when the federal government finally decided to regulate the patent medicine industry, it was these commercialized versions that would be targeted. In October 1962, President John F. Kennedy signed the Kefauver-Harris drug amendments into law. The amendment came in direct response to the thalidomide disaster, a sedative drug that had caused severe birth defects in thousands of children in Europe. Congress wanted stronger controls on pharmaceutical safety. The stated goal was reasonable. Before 1962, drug manufacturers only had to prove their products were safe. After 1962, they also had to prove they were effective, using randomized controlled clinical trials. What followed was one of the largest regulatory sweeps in American history. the FDA launched the Drug Efficacy Study Implementation, known as DESI. Over the next decade, the agency reviewed more than 3,400 drug products that had been on the market since 1938. Thousands were pulled from the market, among them products containing chokecherry bark, echinacea root, and dozens of other plant medicines with roots in Native American pharmacological tradition. But here's where the logic falls apart. The FDA's clinical trial standard was designed to evaluate pharmaceutical drugs, patented compounds, and single molecules synthesized in laboratories. It was never designed to evaluate 2,000 years of empirical observation. And nobody, not the FDA, not Congress, not the pharmaceutical companies lobbying for the new legislation, was particularly interested in funding clinical trials on plants that nobody owned a patent on. So the bar that was set required proof that these products could never afford to generate. And this is where it gets worse. The FDA's enforcement reached commercial products, but it reached the reservation too. Federal Indian Health Service facilities were already underfunded, understaffed, and often hundreds of miles from remote communities. Lakota healers continued doing what they had always done, quietly and off the record, outside the reach of enforcement, but also outside the reach of any formal protection. The 1970s brought a slow change in academic attitude toward indigenous knowledge systems. A new generation of ethnobotanists began approaching Native American plant medicine with genuine scientific curiosity rather than condescension. Researcher Daniel Moerman at University of Michigan began systematically cataloging what indigenous communities across North America had known for millennia. His Native American Ethnobotany database ultimately documented over 44,000 medicinal plant uses across 291 Native American groups. The chokecherry alone had recorded medicinal uses by 66 different indigenous communities, 66 independent groups, spread across thousands of miles of geography, had each converged on the same plant for the same purpose. That is not coincidence. That is clinical evidence, distributed across centuries and communities. When that many separate peoples independently reach the same conclusion over 2,000 years, you're looking at the oldest form of clinical evidence that exists. By the 1990s, preliminary studies on chemical compounds in native plant species were being published, but the pharmaceutical industry remained largely uninterested. No patent meant no profit, and without funding, research moved slowly. That began to change as antimicrobial resistance became a global health crisis. The World Health Organization declared antibiotic resistance one of the greatest threats to human health in 2015. Suddenly, the question of where new antimicrobial compounds might come from became urgent. Researchers started looking in places they had previously dismissed. In 2019, a team of researchers published results in the Journal of Ethnopharmacology that reframed the entire conversation around indigenous plant medicine and antimicrobial activity. The study examined antimicrobial properties of plants documented in the Native American Ethnobotany database, specifically targeting species used by Plains tribes, including the Lakota. The chokecherry and related Prunus species were among those tested against Staphylococcus aureus, one of the most common causes of wound infection. The results were significant. Chokecherry bark extract showed measurable inhibitory activity against Staphylococcus aureus, and the researchers described the antimicrobial effect as statistically significant, with results that warranted further clinical investigation. What made the findings particularly striking was the mechanism. The prunicin compounds in the bark were found to disrupt bacterial cell membrane integrity, a mode of action that differs from most conventional antibiotics. This matters enormously in an era of antibiotic resistance, because bacteria that have evolved to resist one mechanism of attack often remain vulnerable to another. That is not primitive knowledge. That is a pharmacological advantage that the entire field of medicine has been searching for. 2,000 years ago, the Lakota identified a plant that attacks bacteria through a mechanism modern antibiotics cannot replicate. The pharmaceutical industry has spent billions of dollars searching for exactly this kind of compound. The 2019 paper was not the only relevant study. Research published around the same period from the University of Connecticut and from Indigenous health researchers in Canada reached parallel conclusions about the antimicrobial validity of Plains Indian medicinal plants. The convergence of findings was hard to ignore, but what the research cannot answer is this: how many people in the decades between 1962 and 2019 were prescribed ineffective treatments for infections that chokecherry bark might have helped? How many indigenous communities were restricted or dismissed for practicing knowledge that was, all along, scientifically sound? The chokecherry story sits inside a much larger pattern of suppressed indigenous knowledge. The National Institutes of Health estimates that roughly 40% of modern pharmaceuticals derive from natural compounds first identified in traditional medicines. Aspirin came from willow bark, morphine from poppies, and the anti-malarial drug quinine came from Peruvian indigenous knowledge about cinchona bark. What was never documented, never studied, never validated before it disappeared, nobody can say. The Lakota Nation today maintains active efforts to preserve and revitalize traditional healing knowledge. The Lakota Language Consortium documents medicinal plant knowledge alongside linguistic preservation. Pejuta Wicasa continue practicing within their communities, working in parallel with modern medicine. But knowledge that is not transmitted is knowledge that dies. The boarding school era alone interrupted transmission for at least 3 generations. What survived did so because specific individuals made the choice, at personal cost, to remember. A plant used to treat infection for 2,000 years was banned by a government that had never tested it, then vindicated by science 60 years too late. How many others went the same way, and what was lost in between? That is the question nobody in a position of institutional power seems particularly eager to answer. If this story made you question how medical knowledge gets decided, who gets to decide it, and what the cost of that process has been, there is more to explore. The history of suppressed medicine is longer than most people realize, and the Lakota are far from the only people affected by it. That video is on screen now. And if you want more investigative content like this, subscribe. This channel covers the history that institutional sources tend to leave out.